Hello all my A-level lovelies. This is one of 4 PowerPoints on option A, which goes with component 3 of the EDUQAS syllabus that you're doing. Just because I'm doubting we're going to have enough time to make it through the syllabus, I'm recording this one now so you can basically do this in your own time. The parts of the specification that we're covering here are mainly keyword heavy, so all the terminology that goes with the unit initially. Secondly, looking at the human body as a host. And thirdly, we will be going through a couple of very typical infections or diseases, bacterial, viral and protist infections. So that's what we're doing. Throughout your school career, you will already have worked on different ways of grouping and classifying diseases and illnesses. Now, the most commonly used one, a very early one, is the idea of communicable and non-communicable diseases. A typical non-communicable one is osteoporosis, for example. You can't catch bone issues like that come through lifestyle factors by sitting next to a person who's got it. It would be just as insane as thinking that you can catch a broken leg of somebody. However, communicable ones are the ones that you can spread and pass on, obviously like COVID or athlete's foot or tuberculosis as typical examples. Now, you will have had the classification of disease into the type of pathogens that are causing it, viral caused versus bacterial or fungal caused or even protista caused. Or you may have done those quite typical mind maps with how do diseases spread. Do they spread through infected water? Do they spread sexually? Do they infect through blood? Do they infect through food, contaminated food? Or what are the means of transmission? Is it through an animal vector? And so on. So that is something you will already have done. There is a little quiz you could be doing on the EDUQAS website or the WeJEC website, which literally is the starting point of where you should be right now, and kind of do a little cold test and see how much you still remember. I've got these here as links if you're getting this as PowerPoint. I've also got the QR codes, obviously, just in case I'm sharing this through other means. From your component 1 unit on microbes, you should already know that we are a host for various microbes. If you remember rightly, a eukaryotic cell is usually something in the region of 50, 60, if it's a big plant cell, maybe something like 80, 90 micrometers. And a prokaryotic cell is generally in the region of 5, 6, 7 micrometers. So very small in comparison, almost the size of a mitochondria and a chloroplast. Now, obviously, those microbes are living, most of them, in actually quite a friendly communal way with us. Most of those organisms are not harmful to us. However, some are, and these ones are the ones that are causing disease. For example, one bacteria, which is a bit of a weird one, because if it is in our upper gastrointestinal tract, Escherichia coli or E. coli is causing quite severe food poisoning. However, it's absolutely fine and needed in our lower intestinal tract, so the large intestine, where it actually digests fiber that you're consuming and produces vitamin K for us. Our body is host to various other diseases and parasites. Obviously, you've got head lice, you've got intestinal worms and so on, but also we've got skin mites, and they quite naturally eat dead skin cells off the top. They live with us generally in a mutualistic relationship, and there's not much of a problem. However, if we're interfering with that system, let's say by having excessive amounts of makeup that we do not wash off, that can lead to excessive food being available for mites, and they're doing quite well. They're reproducing too nicely, and then we might get issues. We also have protista living on us. Entamoeba are, the video should kind of show you that little GIF that I've got in the top corner. Amoeba move through, moving their cytoskeleton, flow a little bit similar to a white blood cell would flow and engulf pathogens, feeding on dead gum cells, feeding on the bacteria in your gums, and they can reproduce much quicker if your teeth aren't cleaned. Typical poor breath and inflamed gums situation. We have quite a delicate balance of microbes living with one another in our bodies, a gut microbiome that is meant to be as it is, a flora in your mouth, a flora in your genital regions, your private areas, and usually the bacteria cultures in those areas are holding each other in check and everything's under control. However, if this balance is disturbed, a fungus can take over. The usual bacteria kind of usually keep the fungal infections at bay. Now, how do you get rid of bacteria accidentally or the wrong way around? Option one, heavy antibiotics, which obviously by nature are meant to kill bacteria and sometimes as a side effect don't only kill the pathogenic bacteria but also the ones that are good for us. Second option is overwashing. That's particularly one for thrush caused on the genitalia regions, which is usually caused by way too harsh cleaning substances used in the penile or vaginal region. In your sane mind, you would never put bleach on your lady parts and gentleman parts down there. You wouldn't, but because exactly that's what it does, it kills bacteria, and then you're going to have fungi growing there instead. Obviously your normal microbiome can be re-established and that can be fixed again. Other option is that you're playing with moisture or with oxygen levels. Usually that's different types of underwear that can be either causing a lack of oxygen or too much moisture being held in place. So that was the first part. That was looking at the human body as a host. Now, second bit here, we're going to look at a few key diseases. We're looking at cholera, tuberculosis, smallpox, and influenza as a few diseases that really you should know about, particularly in the current context of COVID and however that's going to have an impact on your university careers. The first one, cholera. Now, this is going back to the microbiology unit as well. Cholera is a gram-negative bacteria. What did that mean? Gram-negative have thin peptidoglycan. They over it have a fatty layer, a liposaccharide. My memory aid, gram-negative. We don't like fat, the idea of negative, fat being a negative thing, which blocks antibiotics from reaching the peptidoglycan layer. We'll get later on to how antibiotics actually break the enzyme that is causing the cross-links in that peptidoglycan layer. We'll get to that. Gram-negative. It is not staining and not holding on to the crystal violet stain, which means that with the iodine washing and with the alcohol washing, it will lose the color. The counter stain, the safranin, will later make it pink stroke red. Now, cholera can only reproduce inside the human host. It is a typical water spread disease, though. Cholera is endemic, not pandemic, endemic, which is if you've got a disease occurring frequently in a location or a particular population. We're talking about warm, impoverished, rural areas where either the knowledge is just not there of how cholera spreads, or the financial system to actually just get clean drinking water to people is just not there. If you did have like 3 minutes of your time, watch this video. It's called The Story of Cholera, a lovely graphical way of showing how cholera spreads. It is spread through contaminated food and water. Carriers may not even know they carry the disease, however, they will still spread it and therefore keep spreading and keep spreading and keep spreading, completely unaware that they're doing so. Now, we're talking about feces with the bacteria getting into drinking water or onto food again, which is the main cause. If you are infected and you are symptomatic, the bacteria reproduce inside your digestive system. The toxin that these bacteria produce interfere with the transport of various ions and water in the small intestine. So water and ions cannot be absorbed the way that they should be, which leads to very, very watery diarrhea, and that can happen quite quickly. Within hours you could have diarrhea, bloody diarrhea, your blood pressure will drop, not enough salts in the body, and coma and death can happen within hours. So we're talking about needs of the need to get onto drip or rehydration therapy as quickly as possible, and antibiotics as quickly as possible. Now, we are in a wealthier country, so for us to think, okay, let's go to hospital, get a drip with fluids in and get antibiotics in, that is something that seems natural to us. What do you do if you're in an impoverished rural area? The first one here is the cheaper option, and if cholera is not as severe, may just do. It's either called oral rehydration therapy or oral replacement therapy. The idea is that you're getting the right balance of salts and sugars and waters back into your body. Now, here in the lovely Western world, what we do, we go to the pharmacy and we buy something, a brand name Diorolite, for example, which is literally an isotonic drink. You could as well buy yourself a Lucozade Sports, where you've got sugars, you've got electrolytes. Basically, that's salt and sugar. That is basically what it is. And yes, in the fancier versions, you have got a mixture of different salts. You've got your sodiums, magnesiums, potassiums, chlorides, and so on. You've got all your different salts in there. But what would do is clean water. 1 litre of clean water, can be boiled, can be cleansed with whatever else you want to cleanse it with, chloride tablets. 1 teaspoon of salt to make sure the sodium goes back in. This is all osmosis. And 7 tablespoons of sugar. That's the basic replacement theory. And if you've ever had a fluid like this, the isotonic drinks, they taste a little bit like tears of sweat. You've got the saltiness to go with that. Obviously, in the Western world, we've got antibiotics to treat the bacteria. However, if you get plenty of oral replacement therapy, you can wait until your immune system kicks in, which you will have to do in poorer countries, and just outwait and hope that your body is going to fight the cholera bacteria before it does a number on you. Now, in some countries, the situation is so dire that just the availability of clean water and salt and sugar is already a problem. Can it be prevented? I've taken a picture here of a refugee camp because that is one of the places where in our modern world cholera is spreading like wildfire. Literally the idea of sewage and feces making it back into the drink water supply somehow by some means or another. And if you've got a dense population of refugees without it being able to go toilet anywhere clean, then you're going to have a recipe for disaster. Now, what can you do? Sort out the hygiene situation. It is a bacteria. It doesn't jump magically from one place to another, and it's not airborne as such. So as it is waterborne, foodborne, it is trying to separate the sewage and sanitation area from the water purification, either through huge distances or through sealed off containers and facilities, and just making sure latrines are available. Also, you could vaccinate, you could do it the other way around. However, there are cheap ways to prevent the spread. I would like to take you on a little journey back to the time 1854. This is a map of London. London was one of the places where cholera outbreaks were kind of sporadically every now and then happening. I mean, it could be as simple as a dead rat or some poo in a well. We're talking of days when you get your fresh water from the well, you've got your bucket on a string and you're lowering it down into the well and pulling up. That would be how you're getting it, or you're getting later on, you're getting pumps. Still, if something has fallen into the well, then you will have a spread of cholera. The story is involving John Snow. John Snow is basically the person starting modern epidemiology. So the idea of if a cholera spreads through the air, it was thought to be caused by bad air, and if someone's got it in the air, so it would be spreading through the air. Now, John Snow has suspected it could be the water supply, so how can he prove that it's spreading through water? He mapped the cases in the homes where they are, tried to find the water pump or water pumps later on involved to remove the handle, which means that you cannot actually pump water from that well. And ta-da, the case is dropped. So through that, yes, that house or those homes would have to go a bit further to go and fetch their water, but at least they would not get contaminated water.